Good afternoon. I hope that everyone has had some coffee. I was told earlier that because of my strong focus in superheroes that there'd be some level of coffee drinking game about every time I mention superheroes. So if you're a How I Met Your Mother fan, superhero, so then you know who I'm looking at over there. So let's talk about Abeona Therapeutics to get things started. Of course, we are a publicly traded NASDAQ company, so in order to give you our forward-looking safe harbor statement, I will be discussing some clinical trial results, which in the gene therapy field are, I think, very, very exciting. So one of the things that you heard on many of the panel discussions is that the importance of patient advocacy and the importance of the foundations on how they come in and support many of these rare disease gene therapy groups. And we wouldn't be here today without many of these foundations which came in when we were a private company and helped support us and are now shareholders in the public company. So it's a nice model. It's actually, I think, a great example of one of the few times where you can point back and say, look, all these foundations came in to support the private company, and as a result, when we went through a public transition, are now shareholders and certainly see a return on their initial investment. So to give you a brief overview, we're going to talk just a little bit about one of our lead programs today, or actually our two lead programs for Sanfilippo syndrome type A and also epidermolysis bullosa, the most severe form, which is a butterfly skin syndrome. So to talk about our AAV programs, which are all lysosomal storage diseases. So raise your hands if you know what the lysosome is, right? Anybody? Right, all right. So a couple of people know, I see in the back. The lysosome is the garbage factory of the cell, right? And the garbage factory, they break down sugars and they provide the metabolites to the rest of the cell. Kids born with these, and there are over 70 different rare diseases like this, they can't break down these sugars. And so the lysosomes become action-packed with the sugars, and there's a lot of neurological deficits. So here is the therapy that we're forming.
In which everything is tied together in a logical way. But there are many serious genetic diseases, and Sanfilippo syndrome is one of them. We are developing a therapy to correct the underlying disease, which is injected into the veins to access and treat all tissues of the body, particularly the brain. Sanfilippo is a disease in which the human body is not able to break down sugars, and we've learned how to fix this process inside a specific part of the cell.
That is a virus.
Lysosomes. We use a virus to deliver the correct DNA into each cell, which for the first time will then start to produce the enzyme necessary to break down sugars. Get involved, developing therapies for rare genetic diseases. Abeona, working together to find a cure.
So we all like commercials and animations, so a little bit, that's the 30-second capture for the rest of my talk. So if you got all that, you don't need to pay attention for the rest of it. So we'll keep talking though. But so here's our pipeline. So Abeona is a rare disease gene therapy company. We have 4 programs in the metabolic space. They all use a particular AAV called AAV9. It's the one AAV that's able to cross the blood-brain barrier. And for many of these diseases where the CNS is a profound component of the disease, they also have a number of systemic complications. So we're delivering this for our top 3 programs as an intravenous delivery. And for our 3rd program, we hope it to be one of the first combination deliveries, both for a direct CNS application and systemic. So our goal is to try and shoot with one virus to be able to target all aspects of the disease. And then we have our dermatology program, which is for epidermolysis bullosa. It's a devastating skin disease, and I hope you have a strong stomach, because we do have a couple of pictures to demonstrate just the disease severity, and some other programs in hematology. You'll notice over here on the right-hand side, these check boxes. Okay, these represent the orphan product designations in the FDA and the Europe, and also the rare pediatric disease designation, which from an investor standpoint and certainly from an approval standpoint is certainly very valuable and helpful to move that along. And also fast track designation for the MPS IIIA program, which will allow us our rolling BLA. And you'll see more of these check boxes kind of translate throughout the year. So for our clinical stage program, you heard, of course, that animation talked about what Sanfilippo syndrome is, and it's a lysosomal storage disease. So here you see a normal cell, and over here you see a cell with lysosomal storage deficiency. All of these holes are actually the lysosomes that are action-packed with sugars that are not broken down. So we are focusing on the 2 most common forms, Sanfilippo syndrome type A and type B. Which also code for the SGSH or NAGLU enzymes. And these originally came out of Nationwide Children's Hospital in Columbus, Ohio, which also if you may have heard of another small company called Avexis. It's a very similar therapy using a self-complementary AAV9 to treat a CNS disorder. So we're using something very similar to the Avexis program as well to treat these devastating diseases. And it's important to note as we talk about there are no other approved therapies for these diseases, and 70% of these children with MPS IIIA don't actually reach the age 18. So in order to prepare for clinical trials, the hospital prepared and conducted a natural history study. And I think you've heard a theme throughout much of the investor space and in the public company space, the importance of natural history studies. This helps you evaluate different clinical outcomes as well as understand the disease progression of these patients. Since there are so few patients in these rare and ultra-rare diseases, you don't see a lot of these natural history studies, so it's very hard to actually demonstrate if you demonstrate efficacy. And one of the best examples I think I heard from back in the 90s was that when people are trying to come up with these new gene therapies, the question was great, you're able to show some delivery, but how do you know if it's actually going to work in these patient populations if you don't have an understanding of the disease? So these natural history studies help with that. And we showed that 25 subjects in 3A and 3B through 12 months. We looked through a number of different follow-up endpoints. This is the natural history study data. So again, you can see that these children fall off a cliff between the ages of about 4 and 7, where they have a severe prognosis, but a severe deterioration in many of their neurocognitive scores, both on a nonverbal IQ and in a behavior assessments. We started conducting a phase 1/2 clinical trial at Nationwide Children's Hospital, which is now expanded globally. We've enrolled 3 patients at the low dose cohort and 3 patients in in cohort 2. We now have sites open, and again, we'll talk about this just a little bit later. Primary objective was safety with secondary efficacy endpoints, looking at reductions in urine and CSF gags. Gags are the sugars. When you hear gag, think sugar. And a reduction in liver volume as a modification of disease, and again, a number of neurocognitive assessments. So first and foremost, we want to demonstrate that things are safe. And you hear in gene therapy, a lot of the problems that come up sometimes are against liver responses. So this is really the graph or the slide that shows that in the first 3 subjects in cohort 1 and the 3 subjects in cohort 2, that serum transaminases, ALT, AST has maintained very, very well. Again, you see the ALT in green and the ALT in purple. A little hard to tell with this lighting, but again, you can see these patients originally present with a bit of an elevated ALT that normalizes, so through 6 months. And again, in the cohort 2, things are again looking very, very stable and safe. So we're very pleased to see that safety with LFTs seems to be a good thing in this program. This is really when you talk to and read some of our public reports about this particular gene therapy, this is one of the things that gets a lot of people excited. Again, remember, we're giving an intravenous gene therapy as a virus, right? We're giving an AAV gene therapy. And what we're seeing here is a dose-dependent reduction in the actual sugar in the CSF, so the cerebrospinal fluid. So in cohort 1, in these black bars, you see by day 30 a 25% reduction, and by day 180 about a 60% reduction. Well, when we dose escalate, we're actually seeing that reduction now as early as 30 days. And so again, when you see this as absolute scores, again, you see how these 3 patients in cohort 1 came down it took them 6 months to get there. By cohort 2, we're actually seeing this by day 30. So this is really one of the only gene therapies in the world to demonstrate in a lysosomal storage disease this type of benefit in human populations. So again, not to be underestimated that this is really one of the things, a hallmark, but we believe will be successful for many of our other programs. Because again, if you recall back to our pipeline, we have 3 other programs that look just like this in gene therapy, one of which has an IND for the MPS IIIB program, and we look forward to enrolling patients very, very soon. But wait, there is more. When we look at these CSF, with these gags and total sugars. So over here, again, you see black bars represent cohort 1, green bars represent cohort 2. There's more black bars because they're through more days of follow-up. We just don't have the data for the rest of them yet. But you can see that we're reducing the total amount of gags through day 90 or day 180. Similarly, in the CSF for the specific heparan sulfate, you're also seeing reductions of upwards of 50%. So again, this is, when we start talking about hallmarks, you start talking about registratable endpoints, we see a reduction in the CSF. We also see a reduction in the urine. So this is talking about systemic benefits. And then these patients come in with, through the natural history study, we showed that they had an enlarged liver and spleen as part of the disease progression. Over 100% enlargement that was followed through over 1 year. So again, by day 30 and up through day 180, we're seeing an actual reduction in liver volume to the point where patients are actually losing the liver margins for these diseases. This is believed to be a good clinical endpoint. And similarly for the cohort 2, again, we already see a very similar as day 30. We don't have the day 180 data yet. So again, seeing, again, when we talk about that efficacy triangle where you're talking about biomarkers, biophysical, and then some form of CNS or a 6-minute walk test or something, we're seeing at least on 2 of these, we're seeing improvements. And then going on to the third portion of that third arm, we're looking at CNS benefits. So to walk you through this is a little bit of a complicated slide, but I want to make sure that it kind of the message is taken away. You see these points right here? This is where they came in. The kids came in. These are 3 separate kids. They came in for their natural history study assessment. This is a nonverbal IQ assessment. So they came in at baseline and the natural history study and got scored. At 6 months, okay, they showed some deterioration. At 12 months in the natural history study, they showed significant deterioration. And then by the time they came in to be assessed for the clinical trial, 2 of these patients had essentially passed through the floor of this effect, or the floor of this score. They had deteriorated to the point where they couldn't actually be scored. Okay, even here, this one had gone down to the score. However, 6 months post-injection, these scores had improved. So, we're very hesitant to, claim a lot of efficacy on this, but at least we can tell you that at least in 3 patients, we did see an improvement in the amount of scores that they could be completed. So again, when we look at what would have predicted from the natural history study score would have been a drop in 11 points for this particular subject. However, we're seeing a swing of 10 points, or an improvement in 10 points. So a swing of 20 points towards the positive. So again, we're very excited by that as an early benefit, an early potential benefit. And now that we're enrolling cohort 2 at a higher dose in younger patients, we do think that has a great potential. In gene therapy is all about delivery. And so now as we're trying to treat younger and treat with more, we think that this has a lot of potential, a lot of hope. Similarly, when we look at the Vinland adaptive behavior scales, again, this is an average of 3 patients. You can see that by the Vinland, they would have predicted an average reduction of about 7 points. Over cohort 3, you saw a change of only 3 points. So again, a swing of 4 points, but again, it's in the right direction. So summarizing our MPS IIIA program, we have seen evidence of biopotency and clinical benefit. We're seeing some positive signals in the early neurocognitive assessments, and things are certainly well tolerated to date. I don't have a lot of time left, so I'm going to kind of speed through this next couple of slides. This is really some of the disease paradigms, and happy to talk offline about this a little bit more. But this is what we're hoping to see. This is where we enrolled some patients. You're trying to see disease stabilization or disease attenuation. Now, as we start to enroll younger, you're hoping to see some form of functional cure or even just an attenuation of this, or even at best, disease stabilization. So a lot of things to come in this trial. Now we are also enrolling this overseas in both Spain and Australia. So a lot of excitement on this. And again, recall we have 3 more programs that are using the same type of delivery method for a very similar disease. So gene therapy being all about trying to get it there. We know that we are at least showing in humans some of the first proof of concept. And as kind of a takeaway point, we have enrolled more patients in a gene therapy trial in a lysosomal storage disease than any other company in the world. So I'll let that sink in. And with that, I think I'm out of time and I'll take questions. There's a whole bunch more slides behind this to talk about epidermolysis bullosa. I'll leave you with this and say, this is a patient with epidermolysis bullosa or butterfly skin. Our program is able to see a lot of these wounds close 2 years post-treatment. A horrible disease, and looking forward to sharing more data about this in the next year. So, thank you.